Welcome everyone. In this video, we'll explore the contamination control strategy for sterile manufacturing, or CCS for short. This is one of the most essential frameworks in modern pharmaceutical production. It helps ensure that every product we make is safe, pure, and completely free from contamination. We'll go step by step through its key concepts, from facility design to personal training. So, by the end, you will clearly understand how contamination is prevented before it even begins. So, first, what exactly is a contamination control strategy? In short, CCS is a system that looks at every single source of contamination, whether it's microbial, chemical, or even particulate, and applies risk-based controls to prevent it. It covers the entire manufacturer journey, from raw materials all the way to the final sterile product. In other words, CCS is not just one procedure. It's an integrated mindset that ensures sterility at every stage of production. Let's look at the three core principles behind the CCS. First, control by design. This means we build contamination prevention right into the process using quality by design and risk management. Second, organizational control. Every procedure, every role, and every document is structured under the company's quality system. And finally, continuous evaluation. We don't just set controls once, we constantly analyze trends and review data to catch any early warning signs before problems occur. One thing that makes CCS unique is that it's a living document. That means it's never truly finished. As technologies evolve or new risks appear, the strategy must be updated and improved. We also combine both proactive and reactive elements. Proactive measures like continuous improvement and risk reviews, and reactive ones like CAPA, meaning corrective and preventive actions whenever contamination events occur. In simple terms, CCS keeps learning and growing along with your manufacturing process. Next, let's talk about the physical environment, the facility, and equipment. Clean rooms are the heart of sterile manufacturing. They are designed according to ISO 14644 standards with positive air pressure and HEPA filtration to keep contaminants out. Barrier systems like wraps or isolators are used to minimize direct human contact, and regular qualification tests like airflow visualization and filter integrity checks help ensure every stays under control. In CCS, good design equals good protection. Clean rooms only stay clean when we maintain them properly. That's where cleaning and disinfection programs comes in. We always clean first, then disinfect, using validated chemicals and rotating agents to avoid microbial resistance. Utilities like water for injection, steam, and compressed gases also play a huge role. They must be designed to prevent particulates, biofilm, and pyrogens. In short, cleaning and utilities form the invisible backbone of contamination control. People are often the biggest contamination risk. That's why training and discipline are absolutely crucial. Personnel working in clean rooms must be fully trained, qualified, and regularly re-evaluated. They wear sterile gowns, gloves, masks, and other protective gear with no personal items allowed inside. Beyond procedures, it's about culture, developing awareness that every movement and every touch matters in a sterile area. In aseptic processing, prevention is everything because once contamination happens, there's no statistical way to guarantee sterility. So, we simulate the process using media fields to test aseptic performance under worst case conditions. We also validate sterilization methods, limit open containers times, and confirm container closure integrity. All of these steps work together to ensure that each batch meets the highest possible sterility assurance level. To know whether your system is truly under control, you need strong monitoring. Environmental monitoring tells us if the clean room and process are staying within acceptable limits. We set sampling points based on risk assessments and error visualization studies, and we track results through data trending and alert levels. If trends shift or limits are breached, it triggers an investigation and CAPA. Monitoring doesn't just collect data, it's how we keep the entire system alive and responsive. So, how do we actually build and maintain a CCS? There's a simple three stages approach. Stage 1 is development, identifying contamination risks and defining preventive measures. Stage 2 is compilation, putting together all supporting documents like risk assessments, validations, and SOPs. And stage 3 is evaluation, reviewing results and updating the CCS based on performance data. This cycle repeats under management oversight, ensuring continuous improvement over time. To wrap up, contamination control is not just a checklist, it's a philosophy. A well designed CCS ensures product sterility, safety, and full compliance with GMP Annex 1. It brings together design, operation, and quality culture into one unified system. When done right, it prevents contamination long before it can even reach your product. That's what makes contamination control the true foundation of pharmaceutical